Right. So, good evening, children. Right. So, we'll start the class. Right. So, after about 10 days leave, we resumed the classes again. Uh, right. So, we were in the unit number 4 discussing the plant form and the functions. Right. So, in the first unit, we discussed about the types of tissues that you have. I hope all of you can hear me clearly. If somebody is having any difficulty in hearing this one, you can put a message. Uh, right. So in the first tube today, we discussed about the types of plant tissues, right? About the meristematic tissues and the permanent tissues, right? In the second tube, now we are in the second tube, and in the second tube, we discussed about the primary growth, which increases the length of the shoot system and the root system. And then we discussed about the secondary growth in the dipot plants, where the circumference is increased, right? So we finished those things in the last class, right? So we are in the latter part of the unit tube number 2, right? So here now, after the secondary growth, Pute, now we are focusing. You have to know something about the plant. After the secondary growth of the dicotyledons, you can see different areas of the plant stem, right? Basically, in the shoot system, right? So those are the in woody plants. We consider the dicotyledons and the monocotyledons as the woody plants, and especially the secondary growth happens in the dicot plants. And for the most of the furnitures that we use as wood is taken from this after the secondary growth of the dicot plants, right? So when you may have seen the cross section of these plants after the trees are fallen down for the use as the furniture and the wood for the house construction and various things that woods are used, right? So you use this very tough part of the plant, right? So there are some various types of areas in a plant stem, right? So those are the things that you are going to learn at this latter part of the secondary growth. Okay, right. So in this plant today, there is a thing called hardwood and sapwood, right? So what do you mean by this hardwood? Uh, Hardwood means today, you know, mm, in the secondary growth, you have the uh, cambium, and from the cambium to the inside, you produce the secondary xylem, isn't it? This is the cambium, and to the outside, you produce the secondary phloem, isn't it? This is uh, secondary phloem, and this part, which I mark with these strips, that is the uh, secondary xylem. Okay. Now, when the plants passes years and years, the secondary xylem is collected towards the inside, and the innermost part of this secondary xylem, right? Now, this is the secondary xylem, the older part, right? They are compressed to the inside. This older part, right? I'll mark it in the blue color, right? This is the older part of the secondary xylem. This whole part is the secondary xylem, right? To the inner core, you get the older part of the xylem's secondary xylem. older region of the secondary xylem. Now, what happens? This older part, they are the cavity, cell cavity also filled by various types of resins and other compounds. So the transparency of the xylem is lost in this older part, and they get very tough, right? Compared to the secondary xylem, which is function. So this older part is defunctioning now. They are not functioning. That means they are not transporting the water and minerals. Their lumens are closed with the resins and the other compounds. Therefore, they are closed and that part is very hard part because of the resins and these things. Right. Now, this part is called as hardwood, not the hard, not HARD, HEART, Hadavat, right? That heart. So why you call it hardwood? Because it is located at the center of the stem, right? At the center, at the heart of the stem, at the heart of the shoot system, this wood is uh, uh, located. Therefore, this older part of the secondary xylem is called as the hardwood. Secondary xylem is called as the hard, HEAR, hardwood. Okay, right? So, good evening, good evening. Okay, students, good evening, everyone. Right, we started the class, and so, okay, right. Uh, we have a small part in the second unit. That is what I am finishing after the secondary growth. So last week we discussed about the primary growth and the secondary growth. We finished the secondary growth. And after the secondary growth, there are some areas in the stem of a plant. Those are the things that we are going to learn here. This is a very small part, very few definitions at the cross section of a plant, right? So the first thing that I'm telling again for the students who just joined now. So the first thing that you are going to learn here, two words, heartwood and the sapwood. Heart means this heart, not hard, HEART, heartwood. Heartwood means, okay, now this is the uh, vascular cambium, right? This is the vascular cambium. And vascular cambium, you have these, they are having the lateral meristems, which increases the diameter of the circumference of the plants in the diploid plants. So from the vascular cambium to the inner side, to the cortex, they are making the secondary xylems, right? So from here, they are making the secondary xylem. To outwards, secondary phloem is made. Inward, secondary xylem, right? So focus about the xylem. Now to inwards, they make the secondary xylem, right? So all this area is secondary xylem at the core. Now, towards the center of the stem, more older parts of the secondary xylem is located. Now, this is what I have marked in dark area. Those are the older parts in the secondary xylem. Right? Don't get confused. All this area is secondary xylem. In the secondary xylem, towards the center, you have the more oldest ones. Right? So, once they are becoming more and more older, their lumen, xylem lumen, which transport the water and minerals, they defunctions. Why? Their lumen is also filled with some of the resins and various compounds. Right? Therefore, the transparency is lost. Right? So they are no more transporting the water and the minerals. So, but they are located, as they are located at the center, they are called as the hardwood. Right? Why? They are located at the heart of the stem. That is why it is called as the hardwood. Then the other part of the secondary xylem still transporting the the water and the minerals from the roots to the leaves, right? So there, that other part, the younger part of the secondary xylem, they are still filled with xylem sap. They are still filled with xylem sap. Therefore, that area is called as the sapwood. Why? You have the water and mineral or the xylem sap is still there. Right? So this is the sapwood, this is the heartroot. Understood? Right, very easy, clear. Okay, right. Shall we fill that part in the note? Sir, should we note it? Yes, I am now sharing the note. I hope all of you are having that note. So we'll read it and fill the blanks quickly. Okay? Is that the thing that you asked? Who is that? Your name is not mentioned in iPhone. I can't understand. Identify you. Yeah, okay, okay. Better when you are joining the class to join with your real name, then I know who you are. Okay, right, right. So we'll read that part. Hardwood and sapwood. Right. As a woody plant ages, the older layers of secondary xylem. The older layers of secondary xylem. The older layers of secondary xylem no longer transport water and minerals. Right. These layers are called hardwood. Hardwood. Right. So hardwood, again in other words, today the secondary xylem which do not transport the water and minerals due to the deposition of resins and other compounds are known as the hardwoods. Okay. So these layers are called hardwood because they are close to the center of the stem or the root. Right. The newest outer layers of secondary xylem which still transport xylem sap, which still transport xylem sap, still transport the xylem sap are known as sapwood, are known as sapwood. The hardwood is generally darker than the sapwood because of resins and other compounds that permeate the cell cavities and help to protect the core of the tree from fungi and various wood-boring insects, right? So this is very hard and it is a protective mechanism inside all these fungi and the bacteria. They cannot harm the tree because of this hardwood, right? Only the young secondary phloem function. Now, whatever this is the story about the xylem, no? Right. Look at this one. So this is what happens to the xylem, secondary xylem. So what happens to the secondary phloem? Secondary phloem is deposited and produced outwards from the vascular ganglion. So all the part of the secondary phloem is more towards the outside. So what happens? They just slough off and they are replaced by the newly formed secondary phloem. That is the thing that happens, right? So older region of the secondary xylem is deposited and they become more harder and they are remaining as the very, very tough wood. That is the hardwood, right? The older layers of the secondary phloem just slough off and their duty, the role is replaced by the newly formed secondary phloem. Okay, so that is what it says now. Only the young secondary phloem uh, functions in the phloem translocation, and the older secondary phloem is sloughed off. Right. So I have given you the picture of the hardwood and sapwood, right? Look at this picture, right? Look at this one, right? This dark area, this one is the hardwood, whereas the this area is the sapwood, where the xylem is still functioning, right? So both this hardwood and the sapwood, this whole area is the secondary xylem, right? This is the secondary xylem, but the water and minerals transport happens only in this sapwood area. These are the cell cavities also, the vessel elements and the tracheids all are filled with the resins and the other compounds. Okay, right. Okay, that is the story of the hardwood and sapwood. And another definition is there. Right. So another definition is there. There is a word called hardwood. Now it is HARD, hardwood and softwood. So what do you mean by this hardwood and softwood today? Right? Now hardwood means where the secondary growth happens in the dicots and the in the angiosperms today in dicots the secondary growth happens. And what about the gymnosperms? Do they also undergo the secondary growth? Yes, right. So in gymnosperms, they also undergo the secondary growth. You can see the cypress trees and all these, their circumference and the diameter also increasing with the age, right? But in angiosperms, monocotyledones, they don't increase the circumference, only the dicotyledones increase the circumference. So the secondary growth, in summary, remember, secondary growth occurs only in the dicots in angiosperms and in gymnosperms, right? So the wood secondary xylem, right? Secondary xylem is the hard part of a plant. So what human beings use for the industrial and household things as the furniture and the wood, we use the secondary, basically the secondary xylem part, right? So the secondary xylem in the dicot plants are known as hardwood. That is the part we take as the wood. Again, secondary xylem, the whole, right? Either hardwood or the sapwood, both, right? The secondary xylem of the dicot plants of angiosperms are known as hardwood, right? Secondary xylem of dicots, dicots in angiosperms is hardwood, right? Softwood means the xylem of the secondary xylem of gymnosperms. Secondary xylem of the gymnosperms is known as the softwood. For example, cypress trees, right? And all the other plants of mahogany, teak, jack, mango, right? They are having the hardwood means the secondary xylem of the dicot angiosperms, right? Again, two definitions today, two words, hardwood and softwood. Previously, we learned hardwood and sapwood, that is in the secondary growth of the dicot plants. The functioning part is the sapwood, dead part is the hardwood. Here, all the secondary xylem, the whole part of the secondary xylem of the dicot of the angiosperms is known as the hardwood, and the other part in the gymnosperms, right? Gymnosperms, the plants which undergo the secondary growth, their wood is called as softwood. The difference is here you have the vessels. Can you remember when we learned the tissues in the first tube? We learn what are the cells that are there in the xylem and the phloem. In the xylem, what are the vessels? What are the cells that you have? You have the vessel elements, tracheids, xylem parenchyma, and xylem fibers. Right. So in angiosperms, only vessel elements was there, and in gymnosperms, only the tracheids were there. Right. So the amount of tracheids is less in angiosperms, and more vessel elements are there. Tracheids are less. But in gymnosperms, vessels are absent. Only the tracheids are there. Therefore, this wood is not that much hard compared to the angiosperms. Right? That is why we give this definition. What you have as secondary xylem in the angiosperm is having vessel elements, and therefore they are more hard. Therefore, hardwood is there in the angiosperms. And the secondary xylem that is there in the gymnosperms do not have the vessel elements, only the tracheids are there. They are not harder compared to the angiosperms. Therefore, this one is known as identified as softwood. Understood? Clear? So hardwood, softwood in angiosperms, in gymnosperms. That's all. Okay? Right? Right. Right. Hardwood and softwood. Hardwood is the secondary xylem, of dicot angiosperms. Hardwood is the secondary xylem of dicot angiosperms. Hardwood is the secondary xylem of the dicot angiosperms, while the wood of gymnosperms are named softwood. While the name of wood of the gymnosperms are named softwood. Okay. Xylem vessels are absent in the softwood, right? In the softwood, you don't have the xylem vessels that we learned in the first tube. We need learn the types of cells in the xylem and the phloem. Right? So, hardwood. Wood is tough and heavy with rough appearance. vessels are present today. Vessels are present in hardwood, and therefore tracheids content is less in the hardwood. So in angiosperms, hardwood putte, vessel element content is more, tracheid content is less. That is why it is more tough. Example for hardwood putte, teak, mahogany. Shall I show you the this one, right? Teak, that is one. Mahogany, and jackfruit was jackfruit and rosewood. These are the examples for hardwood. Examples for hardwood. Okay. Right. Softwood. The first point is Pute. Wood of gymnosperms. Softwood is a wood of gymnosperms. Wood of gymnosperms. Wood is slopy and thin with a fine texture. What do you think about the vessels? Vessels are absent, right? Vessels are absent. In gymnosperms, you don't have the vessels. Vessels are absent. And therefore, tracheids content is more in the softwoods, right? Example, cypress. Cypress. Right. So this is the picture that I have given you. This is the gymnosperm plant. So they are having the softwood, and this is the angiosperm dicot plant. They are having the hardwood. Okay, right. Then about the growth rings, there is a right. Excuse me for 2 minutes, huh? Right. Okay. Uh, so then there is another story about this uh, plant stem. Okay. Now, sorry. Now, when the plant growth happens, Pute, during an year, the growth is not having happening in the same pace or the same speed throughout the year, right? So, if you take a year from January to December, right? The growth doesn't happen in the same speed or same strength. There are some rainy seasons and there are some dry seasons, right? So what happens is usually in rainy season where the sunlight is, you are having enough sunlight and you have enough soil water. So the growth is more faster in the wet season, right? I mean, secondary xylem and the secondary phloem is making in very high speed because enough the resources are there. But in dry season, Pute, the growth is very slower because you have a challenge in finding enough water. So you have to protect whatever the water available in the soil solution. So therefore, when you consider the secondary growth of a plant, it is not at a regular speed throughout the year. In wet season, the growth is high, and in dry season, the growth is very slow. And the other thing, not only the speed, Pute, the vessels that are made in the wet season, right? Vessel elements or the xylem, right? They are having thin walls, and larger lumen, right? Large lumen is there. Why? Because water is not a problem at the wet season. So they produce new vessel cells and trachees, which large lumen and thin walls. So more and more water is absorbed from the roots, and there more water is given to the leaves, right, in wet season. But in dry season, right, you have to protect the water. So more water cannot be distributed. Therefore, very little amount of water is given to the leaves and the other parts. Therefore, the new cells produced in the dry season is having a small lumen with thicker cell wall, right? With thicker cell wall, they are making small lumen, right? Thick cell wall, thick wall, and small lumen. Understood? Right. Now, because of these two differences, when you make the secondary xylem, right? The secondary xylem which is made in the wet season is somewhat fair, right? Light in color, right? But in the dry season, the secondary xylem made in the dry season is more darker because of the thick walls, right? Therefore, you make more darker secondary xylem in the dry season. Now, after 1 year, Pute, you can see two colored rings, right? One ring is there, which is produced in the wet season, and it is light in color, and another darker ring, which is produced in the dry season, and it is very dark. So these are called as growth rings, Pute, right? These are called as growth rings. So in temperate countries like our country and Malaysia, right, they don't have these all four seasons. They are having the wet season and the dry season, right? So in the wet season, this and the dry season, these are the two rings that they have, right? But in European countries, the summer, spring, and all these things are there, right? So in the summer is the part where the European countries, their growth is happening in a large scale. Right? Sorry, spring, right? Spring. Summer, you have high temperature is high, light intensity is very high, and it's a dry season in the summer, right? So in the summer, the growth is restricted, and you make the thick-walled, small lumen vessels and the tracheates in the sun. So therefore, sometimes we call them this part as the darker part as the summer ring, and this lighter part as the spring ring. Okay, understood. So these are called as growth rings. So the summer ring and the spring ring is made one ring. The both rings are made only once in a year. So if you have two summer rings and the two spring rings, that means the age of the plant is 2 years. Likewise, now these growth rings can be also used to calculate the age of the plant also after cutting it. So you have to calculate when a tree is cut. You also can count if your plant tree is cut for whatever the usage at your home. So you can go and count these growth rings, and counting these growth rings, you can say the age of that plant. If you have 14, that means 14 years. If you have 55 or 56, that is 55 or 56 years. Years is the age of the plant. Okay, right. So this is the thing that you have to know about the growth rings of a plant. Understood? Okay, right. Mm, mm. Growth rings. Okay, right. The thickness of the secondary xylem and the lumen of vessels are larger in periods of warm and wet seasons. Are larger in periods of warm and wet seasons. Are larger in periods of warm and wet seasons compared to the other growth seasons of the year. Right? These differences are visible in a cross section in lighter and darker rings. Right? So in a cross section, you can see these things as lighter and darker rings. So these are referred as growth rings. These are referred as growth rings. Right? Growth rings. So in temperate regions, wood that develop early in spring is known as spring wood. Is known as spring wood. Spring wood. The xylem tissue, right? The xylem tissue consists of xylem vessels with large lumens and thin walls. Right? So spring wood, large lumen, thin walls, and therefore it is lighter in color. Right? The structure maximizes delivery of water to the new leaves. Right? The wood produced during the rest of the growing season is called the summer wood. That is called as the summer wood. Called the summer wood. Okay. These xylem tissues consist of xylem vessels with thick walls and small lumen. Right? Do not transport much water but provide more support. Okay, they provide more support. Right. These two woods collectively known as an annual ring. So it is an annual ring. Summer wood and the spring wood collectively you call one annual ring. Annual ring. Right? So a year's growth appears as a distinct ring in the cross section of most tree trunks and the roots, right? So most of the tree trunks and the roots, you can see these type of annual rings or the growth rings. Therefore, age of the tree can be estimated by counting annual rings in tree growing in a temperate region. Right. So I have given you a picture also showing these annual rains, right? So rainy season, you have the lighter and more thicker areas, and dry season, you have the darker and thinner areas of the growth. Okay, right. Okay, right. Then the last part of the second tube. Right. So the last part is, Pute, we are going to learn something about the architecture of the plants, right? So usually, plants are the ones that the photosynthesis is performing, right? So they are the ones who gives the energy to the whole biosphere, right? I told you, without water there is no life on Earth, and without the plants or without the photosynthesis, there is no life on Earth. So all the organisms get the energy from the process called photosynthesis, right? So these plants are adapted to get now from in the story of evolution, Pute, the plants invaded the land area. So once the plants are invaded the land area, they have some challenges, right? So basic two challenges or the basic two essential requirements for the plants are the water in the land, water and the sunlight, right? So their basic architecture is evolved to get more and more water from the ground and to get more and more light from the atmosphere. So that is why the plants do not like to grow in the shaded area, right? Because they need sunlight for the generation of the production of the energy, right? So that is why the plants are growing higher and higher and to get the maximum light from the sunlight, right? So the angiosperms, they are having very hard stem producing the secondary xylem, and they are not easily fragile. So they have the hard stem and they go grow meters, hundreds, sometimes 100 meters, seeking for more and more sunlight. So jackfruit, mahogany, mango, whatever the coconut, all these things. Now some are branching, some are unbranched, right? So basically the monocots, they are not branched, but dicots are branching, right? Either branched or unbranched, they grow taller to get maximum amount of sunlight. right? That is about the stem. Then about the leaves, Pute, right? So their leaves also, right? The leaf size is also different depending on the plants, right? Now, the plants which grow in wet season, wet areas, right? For example, rainforest, right? The plants that grows in the rainforest, they are having very large leaves because water is not a problem. Transpiration is not a problem. They have to capture more and more sunlight, so their size is larger. Whereas the plant which grows in very dry and very cold areas, they are having very small or thin leaves because water is a problem for them. So they have to protect whatever the water available. So therefore, to minimize the transpiration, their leaf size is very small, right? And there is another thing that you have to know from one node of a plant. Sometimes in some plants you get only one leaf, and sometimes you get only two leaves, and there are some plants where you for you get more than four leaves from the same node, right? So those are something about the architecture of the plants, right? Just to have a basic idea about the leaf size, and then this is called phyllotaxy. Phyllotaxy means how the leaves are arranged in one node, right? So have a basic idea about these things. Okay, right. Shall we read that part? So shoot architecture and the light capture. The stem and the branching patterns are designed to capture maximum light, right? Plants grow tall to avoid shading from the neighboring plants, right? This is about the stem. So most tall plants have thick stem with strong mechanical support, and woody plants undergo secondary growth, thereby make their tall stem very stronger, right? So vines rely on other objects. So vines also need their light for their photosynthesis. So they don't have a very strong and hard stem. So what they do, they rely on the strong stems and they surround and grows to a taller height and get the maximum light, right? So vines rely on other objects to reach the higher levels to capture more and more light. So that is about the stem. Then about the branching patterns, Pute. There is a variety in branching patterns. Some plants are unbranched, like coconut and arecanut, and still others are well branched, right? So this variation in branching patterns enables the plant to absorb maximum light in the ecological niche it occupies, right? Okay, so that is about the branching. Then about the leaf size and the phyllotaxy. Leaf size, I told you, right? The size of the leaf vary based on the place where the plant grows, right? So larger the leaves are found in plants growing in rainforest, right? So their water is not a challenge, right? Not a challenge for them. So therefore, enough water is there in the rainforests. So the plants that grow in those areas have a larger uh, leaves to do the perform the photosynthesis in a higher rate, right? And smallest leaves are found in plant species inhabiting in dry and very cold environments. Okay, so just have an idea about that those things. Where some only MCQs can come from these topics, right? And the other thing, so Jalani also asked, we missed our sometimes we were continuing some question and answer part at the last half an hour. So we'll continue with it with the next day, right? I I do not think that we can continue from the very beginning, but from the uh, third unit, I'll give you. I'll make some questions and I'll give you, and we'll have a question and answer session also. Sorry, it's this one, right? So what I was talking about the question and answer session of our normal theory class, we missed that thing because of the conversion of the theory class into the online class, and I had to for a few months I have to uh, come to another country also now. Last week I came to Malaysia and now I am doing the class in Malaysia for a few months. I am staying here and coming back. And so because of this transformation, transit and all these things, we miss the normal question and answer session also. So we'll start that it from the next class, right? So we'll have 3.5 a session per one day, and for 3 hours we'll do the theory part, and for last half an hour or 1 hour we'll take for discussing the questions and answers. That is very important for you, right? So we'll start that normal pattern from next day, and you know the uh, classes for this month, and as far as I can remember, I have arranged 5 classes for you, 5 or 4. Uh, So every Wednesday you will have your normal class. So yes, we have scheduled 5 classes for this month, and next week, next month also we'll do some additional classes and try to recap what we have lost because of not having the classes for few weeks. So today is 13 we are having the class, and next Wednesday, every Wednesday we are having the classes, the 30th, 20, 13th, 20, 27. And also we are having the class on 21st and 28th, right? So these are the right. So these are the days in this month, May, that we are going to do the classes. So this is today we are doing this one, and I have shared this schedule with you in the group. Okay, right. Subi and sir, wines means easy to plant. Yes, yes, yes. Subi, uh, wines means in Sinhala we call it well කියලා කියන්නේ, right? I don't know the Tamil word for that. I draw it and show the very flexible. Now these are the large trees that you have, right? And there are some very flexible vines which grow surrounding the plants, and their leaves are there, right? These are called as vines. Okay, well, here, did you got it? Charani. Okay, right. Thank you. Okay. Subi, did you understand that? Okay, right. Okay, okay. Ask your mother also. What do you call it? These are the very flexible, small, the spring-like things that grows surrounding the trees. Those are the vines, right? Trees and vines. There was a poem in the uh, kindergartens also that you are singing for the vines. We say Kodi. Right, right. In Tamil, you call it Kodi, right? So, in Sinhala, you call it well, right? So, wines are Kodi, huh? Good, thank you. Okay. Right. So, that is about the leaf size. Then, about the phyllotaxy means how the leaves are arranged surrounding the plant stem, right? This is the arrangement of leaves on the stem, and the arrangement may be one leaf, two leaves, or several leaves per node. Right, so phyllotaxy helps the plant to capture maximum sunlight. So these phyllotaxy or the arrangement of the leaves are also have arranged to get or capture maximum sunlight. So I have given you a picture about the types of phyllotaxy. You have the opposite pattern, alternative pattern, world pattern. Likewise, in various ways, the leaves are arranged. Right. So remember the stem, branching, leaf size, and leaf pattern. All these things are arranged to get the maximum sunlight, right, for the photosynthesis in the wet season. And if it is in the dry season, right, not the dry season, dry areas, if the plants are growing in dry areas, their leaf size is smaller. And there is another thing about the orientation, right? Look at this one. Now, orientation of the leaves, that means how the leaves are oriented in their plants. Right. Leaf orientation, right? So basically, try to get this one. Uh, if the plants are growing in wet areas like rainforest, Pute, the leaves are oriented horizontally, right? So you can get the maximum sunlight, right? But if the plants are growing in the dry season, where you don't have enough water, so their leaves are vertically placed, vertically. So that is to avoid the sunlight. So you don't get direct sunlight. You can avoid the sunlight. Examples are grasses. Right? So grasses are usually grown in very dry areas, and they are exposed to direct sunlight, and lesser amount of water is available in the soil. So they have to protect water. So their leaves are oriented vertically. Okay, right, right. So leaf orientation, Pute. Leaf orientation. Leaves may be horizontally oriented. They capture light efficiently in low light conditions if they are oriented in the horizontal level. Some plants have vertically arranged leaves, for example, grasses. This is to avoid the possible damage caused by the exposure of the leaf to the over intense sunlight, right? So when leaves are nearly vertical, the light rays are parallel to the leaf surfaces, so no leaves receive too much light, and therefore damage is very lesser. Okay, right. So I have given you a picture also. showing the vertical and the horizontal orientation of the leaves. Okay, right. So that's the end of the tube number 2, right? So growth and development of the tissues is finished, right? So in the unit number 4, plant form and function, we finished the first unit that describes everything about the plant tissues, including the meristems and the permanent tissues. And then second tube covered the primary growth and the secondary growth, and about the sapwood, heartwood, hardwood, softwood, growth rings, springwood, and summerwood, and the orientation of the leaves, the leaf size, and the stem and the branching and all these things. Okay, right. Any questions up to now in unit number 4? I'll give you 2 minutes while I clean my whiteboard. If you have any questions, you can drop a message. Mm. Mm. Right. So any questions, Kutel? No. Right. Shall we move to the new tute? Right. Tute number 3. Right. I hope all of you have got the tute. All right. So the new tute. Right. So in this tute today, we are going to cover basically about uh, 3 things. Right. The first thing is you are going to learn about the gaseous exchange in plants. Gaseous exchange means, right? So gaseous exchange happens, Pute, in every bioorganism, right? We also perform the gaseous exchange. What is that gaseous exchange, right? We absorb oxygen and release the carbon dioxide, right? So plants also having a gaseous exchange. What they do is they absorb carbon dioxide, which are used for the photosynthesis, and they release the oxygen to the atmosphere. So that gas exchange at the plants, you are going to learn how it happens through the stomata. Then about the water absorption of the basically the acquisition of water. Acquisition of water, right? These are the 2 topics that you are going to learn. In the acquisition of water, you are going to learn how the water and the minerals are absorbed and distributed throughout the plant, right? In the xylem. And one of the very important physical property that the cells are having is that water potential. Water potential is important for the water acquisition. That means the absorption of the water and minerals and the distribution of that water and minerals throughout the plant. Right. That whole topic is the oxygen. For that, water potential is very, very important. Water potential. Okay, right. So these are the things that you are going to cover up in this tube number 3. Okay, good. Right. So first we'll learn what is this gaseous exchange business. Right. Right. Okay. So now to understand the gas exchange, Uday, gas exchange means now photosynthesis happens in the plants now, right? So for where the photosynthesis happens in a plant, that is the greenish area, the green area, those are the basically the leaves, right? So leaves are the places where the gas exchange happens. Okay, right. Now in angiosperms today, you have the dicots and monocots, monocotyledon plants and dicotyledon plants. And basic structure of the anatomy of the leaf is similar, but there are some differences in between the monocots and the dicots, right? So first of all, before you learn about the gaseous exchange, you have to know about the anatomy of the plant leaf. Now, this is the second time that we learn. I hope that you remember we learned the anatomy of the leaf in a previous section also. Can you remember what section? In unit number 2, when we learn the photosynthesis, I draw the anatomy and told you about the anatomy of the leaf in C3 plants. And also when you learn the C4 mechanism, we learned about the anatomy of the Kranz anatomy, right? Kranz anatomy in the C4 plants also, right? Again, we'll refresh our knowledge about the plant anatomy. So here we'll first learn about the anatomy of the leaves, and then we'll learn about the gas exchange, how it happens. Okay, right. Right. So Ute, in this is the first we'll get a basic idea about the anatomy of the leaf of a dipot plant, right? So you know you have the upper epidermis and the lower epidermis. This is the upper epidermis today. And then you have the lower epidermis. And in uh, dicot plants, the stomatas, the stomata is the opening of the leaves that is, this opening is there for the gas exchange. So in the dicot plants, stomata are basically located only in the lower epidermis, not in the upper epidermis, right? So this is the stomata. And these are the guard cells that make the stomata. And outside the upper epidermis and lower epidermis, you have a waxy layer called as the cuticle. So cuticle is located here, right? And in between the upper epidermis and the lower epidermis today, you have the ground tissue here. Now you know the structure of the plant, right? You have the dermal tissue with vascular tissue, and in between the dermal tissue and the vascular tissue, you have the ground tissue, right? So here also you have the vascular tissue, right? Those are the veins. You call it veins are supplied in the plants, leaves, right? In a leaf, you can see the xylem and the phloem. The vascular bundles are going as a vein, right? When we are drawing a leaf, now these are the veins that we drew, like a network here in the dicotyledons, right? These are the veins. Veins means you have the xylem plus phloem. Okay. Now that is located here. You have the xylem here and the phloem here. And that vascular bundle is covered by a special type of cells. Can you remember that cells? Which we needed them in the C4 mechanism, right? Covering this vascular bundle, you have the bundle sheath. Those cells are called as bundle sheath cells, isn't it? These are the bundle sheath cells. Bundle sheath cells, right? So this is the vascular tissue. This is the dermal tissue. And in between the dermal tissue and the vascular tissue, you have the ground tissue. Now here the ground tissue is made by basically a cell type called as mesophils. Now mesophils, the name is given for this one, but basically now ground tissue is made by various types of cells. No, you have the parenchyma cells, sclerenchyma cells, likewise. So these are the parenchyma cells, which are living cells with only the primary cell wall, right? So the parenchyma cells, which are located in the leaves, are called as mesophil cells, a specific name, right? But the type of cells are parenchyma cells. Okay. Now, the mesophil cells that are located here, there are two types of mesophils. One type of mesophil cells are linear, right? They are linear cells and tightly packed. Those are called as palisade mesophils. These are palisade mesophils. Palisade mesophils. And the other type of mesophil cells are called as spongy mesophils, which they are spherical and loosely arranged with lot of intercellular spaces. Right? Like this. These are spongy mesophils, loosely arranged with lot of intercellular spaces. Now, photosynthesis happens in palisade mesophils as well as the mesophil cells, the spongy mesophil cells, and chlorophylls are more concentrated in the palisade mesophil cells. And usually the upper epidermis and lower epidermis cells, epidermal cells, you have single cell layer which is tightly packed without any intercellular spaces. They don't have the chlorophylls, but remember the guard cells which make the stomata, they are having the chlorophylls or the chloroplasts, right? Okay, so this is the arrangement of the dicot plant leaf. And I will name this one. This is the cuticle, and this is the upper epidermis. This is the palisade mesophil. This is spongy mesophil. This is lower epidermis. This is cuticle again. These are guard cells. Guard cells. And this is the stomata. Right? So the gas exchange happens from this stomata. Stomata is the opening, right? That opening is created by a bean-shaped cells called guard cells. So guard cells also having the chloroplasts here, and so this is the arrangement of the uh, dicotyledon leaf plant, right? Okay. Shall we come up to this point in the dude? Right. So the process of gas exchange in plants, anatomy of a typical dicot and monocot leaves, right? In most vascular plants, leaves are the main photosynthetic organs. Leaves are the main photosynthetic organs, right? Leaves are the main photosynthetic organs. The exchange of gases occurs through what? Occurs through stomata. Occurs through stomata in the upper and lower epidermis. Right? Epidermis is usually a single cell layer, and in between the upper and lower epidermis, there is a ground tissue called the mesophyll. 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 This tissue consists of parenchyma cells specialized for photosynthesis. So these parenchyma cells specialized for photosynthesis and located in the leaves are called as mesophills. Okay, right. Now we'll focus on dicot leaves, Pute. Right? In dicot leaves, stomata are mainly found in the lower epidermis. Now remember, in dicot leaves, stomata are found only in the lower epidermis. So dicot leaves, stomata are mainly found in the lower epidermis. Lower epidermis. Right. The mesophyll consists of two distinct layers called palisade mesophills and spongy mesophills. Palisade mesophyll and spongy mesophyll. Palisade mesophyll and spongy mesophyll. Right. So palisade mesophyll consists of elongated cells that are arranged in one or more layers. Right. This can be found in the upper part of the leaf, just beneath the upper epidermis. This can be found in the upper part of the leaf, just beneath the upper epidermis. The spongy mesophyll can be found between the palisade layer, and lower epidermis. Between the palisade layer and lower epidermis. Palisade layer and the lower epidermis. So they are loosely arranged with many air spaces. Spongy mesophyll cells have less chloroplasts than in palisade mesophyll cells. Right. So I have just remarked the C4 and C3 mechanism. So If you have time, go back and or else recall the mechanism of C3 and C4 mechanism. Photosynthesis is a very, very popular topic in biology, right? So you recall it and then it is easier for you to understand this anatomy, right? Okay. The vascular tissue of the leaf is continuous with the vascular tissues of the stem, right? And veins of the leaf is highly branched, net-like veination in the mesophyll layer, right? So in dicot leaves, Pute, you have a net-like veination. It's like a network. It looks like a network, right? So therefore, you call it net-like veination, highly branched. Whereas you will understand in the next step when you learn the monocot leaves, they are not branched; they are parallel veins, right? So xylem and the phloem, the veins means to the vascular bundles which consist of xylem and the phloem, right? So in dicot, it is net-like veination that is highly branched, right? So veins in the leaf is highly branched within bracket net-like venation in the mesophyll layer, right? Each vein is protected by a bundle sheath layer. Bundle sheath layer. You remember the bundle sheath cells, which was very important in C4 mechanism, right? So now I have given you the picture because you don't have enough time to draw this one. So uh, you have to name the picture by looking at my drawing. Okay, now see, I hope you can see this one, right? So here you have the cuticle, you have the upper epidermis and the lower epidermis, and in between that you have the mesophyll cells. Mesophyll cells are the two types of mesophyll cells, palisade mesophills and the spongy mesophyll. Palisade mesophills are linear and they are ring shaped and they are tightly packed. You may have one or two palisade mesophyll lines. I have drawn only one. And in the center, you have the xylem and the phloem, so you have to make the same also. This is xylem, this is phloem, and this is bundle sheath. Bundle sheath, right? And you have the waxy layer outside the upper epidermis and the lower epidermis, that is the cuticle. And then you have the stomata. In dicots, you have only the stomata is only placed in the lower epidermis. And these are the guard cells which make that stomatal opening. Right. So quickly you mark these things in your tute drawing. Okay. The only difference is there is a mild change on your right hand side. The upper arrow should mark the upper epidermis. That arrow has come down. So the first one is showing the upper epidermis. The second arrow is showing the palisade mesophills. The other arrows are correct, right? So you mark it. Right. Okay. You finished marking. Right. I'll show you in this one also. Right. So look at the screen that I'm sharing. So this is what? This is the xylem and this is the phloem. Look at my cursor. So this one, this arrow should mark here, somewhere here. That is the upper epidermis. This one is the palisade mesophyll. This one is the spongy mesophyll. And this is the lower epidermis. And this is the stomata. This is the stomata. This is phloem. And this is the bundle sheath covering the vascular bundle. And these are the guard cells, right, which make the stomatal opening. Okay, right. Then the monocot leaves. So we discussed about this thing, right? So we'll complete the note in the monocot leaves. Uh, Before that, you look at the screen. I'll quickly show you what are the differences in the monocot leaves, right? So one thing is in monocot leaves, Pute, you have the stomata in upper epidermis also, right? So in upper epidermis also, I will not draw a new one because it takes some time. So I'll do some corrections here. So here also they are having the stomata, right? So these are the guard cells. Stomata are there in the upper epidermis as well as in the lower epidermis. And the other important thing about the mesophyll cells, right? So they don't have the palisade mesophills, right? They are having only the spongy mesophills. They are having only the spongy mesophills. So spongy mesophills, uh, they have the chloroplasts. Right? So these are the differences in the monocot leaf, right? Okay, right. And the arrangement of veins now in, I told you in dicot plant, the veins are arranged as a net-like venation is there. Now in these ones, just imagine the coconut leaf, right? So these are strong, thin, linear ones, and the veins are parallel like this, right? So veins are parallelly arranged. Okay, right. Mm, just a quick description about the monocot leaves in the note, Pute. So monocot leaves, in monocot leaves, stomata are present in both lower and upper epidermis, present in both lower and upper epidermis. Right? Mesophyll is not differentiated into, it is not differentiated into palisade and spongy layers. It is not differentiated into palisade and spongy layers. Chloroplasts are abundant in all mesophyll cells, right? And veins are parallelly arranged, or you call it parallel venation. Parallel venation, right? Okay, right. So shall we quickly name these parts in your book, in your tute? Shall I name it in the same screen, right, by using my cursor, right? So this is again up now. Look at my cursor. This is the stomata in the upper epidermis, right? In this arrow, this is again stomata in the lower epidermis, and this one is cuticle. This one is cuticle. And this one is upper epidermis, upper epidermis. And this arrow is mesophyll cells. You just write mesophyll cells because you don't see spongy or palisade ones, right? Mesophyll cells. And this is the lower epidermis, lower epidermis. This is stomata again, right? And this is the guard cells, guard cells, guard cells, which make the stomata, stomatal opening. And these are the bundle sheath. This is the bundle sheath or bundle sheath cells. And this is xylem. This is xylem. And they have not put an arrow to the phloem. So put an arrow somewhere here, right, to this area. This is the phloem, right? This is the phloem. So put an arrow to this area and mark it as phloem. This is phloem. Okay. And I have given you the pictures of the monocot leaf and the dicot leaf for your further reference, right? So try to close these. Now, why I am giving these pictures? These pictures can be given in the exam paper. So you close these names and see and check whether you can correctly name the parts of these monocot and dicot leaves. Okay, right. Then the structure of the stomata. Right now, structure of the stomata and uh, opening and closing of the stomata, you have to learn, right? How the structure is made by the two guard cells and how these opening and closing of the stomata happens. You have to know the mechanism, right? So now it's Oh, my watch says the Sri Lankan time correctly now. I hope it is a 7:35, right? I'm still keeping the watch to check the Sri Lankan time. So shall we take a 15 minutes break, right? Now 35 and we will start you take a break and we will start the class in 7:50, right? 7:50 we'll start the class and we'll then continue with the stomatal opening and closing and the mechanisms. Okay, right. A question has asked, oh no, Jalni. Okay, okay, right. Thank you. Right. Right. So we'll start again. Right. Right. So now we are going to start learning now what we learned about the structure of the or the anatomy of the leaf. Right. In this anatomy, you learn where the stomata is located. Right. So in dicots today, stomata is located only in the lower epidermis, and in monocots, it is located in the upper epidermis and the lower epidermis both. Right. So it is made by the guard cells. Right. Now, what you saw up to now is the cross section of the leaf and a small part of the side view of the guard cells. Now we learn the full view of the guard cells and the stomata and their structure. Right. Then, so here you are going to start to learn the structure of the stomata, which is made by the guard cells, and how it opens and closes. What is the mechanism runs behind this opening and closing of the stomata? So those are the two things that you are going to learn. First, we will learn the structure of the stomata, right? Basically, very simple, right? So you have two guard cells. Two guard cells. These guard cells are bean shaped, right? So, like a bean, right? So these are the two guard cells, and they have the chloroplasts, right? These are the only epidermal cells. These are epidermal cells, right? Epidermal cells, specified, adapted epidermal cells, right? So these are the only epidermal cells that have the chloroplasts, right? So all the other epidermal cells, they do not have the chloroplasts, so they are not green in color, but these are green in color, right? And important thing is about the shape, being shaped, you have the chloroplast. Then the other thing is about the cell wall thickness, right? Now, their cell wall thickness is not even throughout the cell. This is uneven thickness is there. What is the unevenness? The inner cell wall towards the pore is somewhat thickened. It is thickened like this. Whereas the outer cell wall is very thin. Right? Inner cell wall is thick. Outer cell wall is thinner. Right? And this is covered by the other normal epidermal cells. Right? So these are the other epidermal cells arranged very tightly. Right? So these are the epidermal cells. These are the guard cells. Right? This is the thickened inner wall. Thinner outer wall. This is the stomatal opening or stomata. Right? These are the chloroplasts. Right? So this is the structure of the stomata. This is the structure of the stomata. Okay. Right. Shall we read the note? If you have any doubt, places, so if you need any repetition, more elaboration, you just put a message, okay? Right. So in the note, the structure of the stomata, right? So stomata are pores surrounded by guard cells. Surrounded by guard cells, Pute. Surrounded by guard cells in the epidermis of the leaves and stems of the plants, which can open and close. So this one can open and close, right? So guard cells are modified epidermal cells which have a distinct shape and are the only epidermal cells that contain chloroplasts. Those are the only epidermal cells that contains the chloroplasts. Guard cells are typically bean-shaped in angiosperms, and the guard cell walls are unevenly thickened. The inner cellular wall is thicker. Inner cellular wall is thicker and less elastic. It is not elastic, Pute. It is thicker and less elastic. It is thicker and less elastic than the outer wall. Than the outer wall. Some of the cellulose microfibrils are radially arranged to form inelastic hoops around the guard cells. Around the guard cells. Right. So I'll explain that part. Now you have cellulose microfibrils. That is like this. Right. Now these are the thicker inner wall now. Right. So from that inner wall to the microfibrils, those are like ropes, right? Ropes which covers the each the inner thick wall is hooked around the thinner out wall by a loop of microfibrils made up of cellulose. This is very important when you learn the mechanism of opening and closing. These are microfibrils made as hoop, right? Likewise, you have some hoops made up of microfibrils, very important for the mechanism of opening and closing, right? Again, we'll go to the somebody else, right? Okay. Manudi has asked a question. So, are the epidermal cells arranged as a ring around the stomata? Yes, yes. Now this is what we see in uh, the horizontal view, right? Not the cross section. That what I have drawn here, Pute, is the uh, right. So take this on. Assume that this is the leaf, Pute, right? So this is the leaf. So if you cut this one and look at the structure here from this angle is what we draw earlier as the anatomy of uh, the leaf, right? So what you see from this shape when you cut this one, right? Without cutting, right? So what you see when you look at the underline of the leaf from this side, right? This is what you can see. Okay, Panti? Right. So all these, so here all are the epidermal cells arranged as a thin single layer, right? So all the epidermal cells are arranged like a sheet here, right? This is the leaf. So we are looking at the back side of the leaf. That is why we see the stomata like this, right? So assume that this board is the leaf. So we are looking at the lower side, right? This is the lower epidermis, and mesophyll is cells are they are behind the sheet, behind the black whiteboard, right? And the upper epidermis is they are in the wall, likewise. Okay, so this is what you see when you look at the underside of the leaf, right? So all these, other than the guard cells and the opening, these all other areas of the leaf is covered by the low epidermal cells. Okay, understood, right? Okay, right. So some of the cellulose microfibrils are radially arranged to form the inelastic hoops around the guard cells. And guard cells regulate the diameter of the stomata. Guard cells regulate the diameter of the stomata. Guard cells regulate the diameter of the stomata by changing shape, widening or narrowing the gap between the pair of guard cells. Okay, right. So I have given you the picture of the guard cells when you see at the under surface of the leaf. Right? Okay. Understood. Any other questions, Pute? Everyone understood? Right. Shall we move on to the gas exchange then? So next, now you know the structure of the stomata, how the stomata is made. Stomata is the opening, huh? How this stomata is made by the guard cells. Now, this is the place where the gas exchange happens, Puti. Right? Gas exchange happens through this opening called stomata. Right? So, stomata gives the way or the pathway to the interior of the leaf. Right? It opens the interior of the leaf to the outside. Right? Can you remember the anatomy of the leaf? You have the upper epidermis, lower epidermis, and in between those two, like a sandwich, you have the inner mesophyll cells, and mesophyll cells are the ones who perform the photosynthesis, and they are rich with chloroplasts and chlorophylls. So photosynthesis means they need the carbon dioxide for the photosynthesis, and as a byproduct, end product, you produce the oxygen. So you need to absorb carbon dioxide to the mesophyll cells, and you have to release the oxygen to the atmosphere. So this is the gas exchange that happens in a plant, right? That's all. Again, right? Gas exchange happens in every organism, right? Animals, fungi, bacteria, plants, everyone, there's the gas exchange, right? Gas exchange means you absorb one gas and release another gas. Now, when you consider the animals like us, we absorb oxygen and exhale carbon dioxide. Why? For the aerobic respiration, oxygen is needed. As a byproduct, carbon dioxide is produced. So we want to absorb oxygen for the production of ATP, and the produced carbon dioxide has to be released. So to the atmosphere, we release the carbon dioxide. So that is the gas exchange that happens within us. Now we are learning about the plant form and function. No? So we are focused on plants, right? In the leaves today, they are also very important gas exchange happens. Now this is the importance of this gas exchange. Because of this gas exchange, we inhale oxygen, right? For thanks to the photosynthesis, thanks to the plants, thanks to the sunlight, we get the oxygen. So what happens? Photosynthesis, they need the carbon is taken from the carbon dioxide. So you have to take carbon dioxide to the place where the photosynthesis happens. Where the photosynthesis happens is at the mesophyll cells. So inside the mesophyll cells, you have the chloroplasts. Inside the chloroplast, you have the thylakoids and all these things are there. The chloroplasts are located in photosystems, photosystem 2 and photosystem 1. So that is the place where the photosynthesis happens. To that place, you need carbon dioxide. So stomata is the main door, which opens the mesophills and allow the atmospheric carbon dioxide to go inside, enter into the leaves and reach the mesophills. And that is the main door which allow the produced oxygen in the mesophyll cells to go out. So this is the main door, stomata, which happens the gas exchange. So gas exchange in the leaves means absorption of the carbon dioxide sent to the mesophyll cells, and oxygen is sent back to the atmosphere as a byproduct. So this exchange is allowed by the stomata. That is the gas exchange, right? Now, what is the mechanism of this opening and closing of the stomata? Very, very important, very easy, very simple. Listen carefully and understand by itself now itself, right? Now look at this one. So I am drawing again the guard cells. Right? These are the guard cells. And I told you that inner wall is thickened and outer wall is very thin. Right? This is the guard cells. Now, how it opens and closes? Very simple thing. Right? You call it this potassium influx hypothesis. You call this potassium ion influx hypothesis. Right now, listen this carefully. What happens today in the morning, in the daytime? Now, photosynthesis happens when the sunlight is available. Sunlight is available in the daytime. So, gas exchange need to happen in the daytime, right? Where the photosynthesis happens, right? So, in the morning to the night, the stomata should be kept opened, right? That should be kept open. And at night, you don't need much of the gas exchange. So therefore, it's okay to close it. So how this opening and closing happens? Now what happens in the morning Pute, the potassium ions are absorbed into the guard cells from the surrounding cells. Now what are the surrounding cells Pute? Surrounding the guard cells, you have the other epidermal cells, Now, those epidermal cells have potassium, and those potassiums are absorbed into the guard cells actively by active transport. That means you spend ATP and absorb potassium. Now, when you absorb potassium into the guard cells, two guard cells, what happens? The solute concentration increases, right, in the guard cells compared to the surrounding cells. When the solute concentration is increased, what happens? The water potential decreases, and therefore water also tends to come into the guard cells because the water concentration goes down here, or the water potential goes down, right? So water also diffused into the cells passively. For that, you don't need energy, right? Because water concentration higher outside, lesser inside, from high concentration to the lesser concentration, water absorbs. You call it diffusion, right? Or osmosis, right? From the osmosis, water is absorbed. Now, what happens in the morning? So, by spending energy, actively potassium is taken into the gut cells, and therefore solute concentration increases, water potential goes down, therefore water also absorbed from the surrounding epidermal cells. Now what happens? The guard cells are swelling, right? You are extending the guard cells, right? The size is expanding like this. So when you expand, now this expansion or extension doesn't happen evenly. එක සමානව වෙන්නේ නැහැ. Why? Because the thickness of the cell wall is different. Inner cell wall are thicker and outer cell wall are thinner. Therefore, expansion happens. What happens? Not much of the expansion here, right? Now, what happens? The curvature of the outer wall increases. Right? The curvature expands like this. This one expand like this, right? And I told you that you have some hoops of microfibrils like this. So when the outer wall, inner wall is expanding like this, from these microfibrils, they drag the inner wall to the outside like this. Therefore, the stomata opens like this, right? Stomata opens. The inner non-elastic thick wall is pulled outwards by the thinner wall because of the expansion due to water absorption. This is called potassium influx hypothesis. Very easy. Understood? Right. Again, I tell you the summary. Right. Understand very clearly at the class. Right. This is called, now what we are explaining, the stomatal opening and closing, the mechanism behind that. One hypothesis that explain that thing is the potassium influx hypothesis. So what happens in the morning? Potassium ions are absorbed into the two guard cells from the surrounding epidermal cells. So what happens? The potassium concentration inside the guard cells increases. Therefore, water concentration goes down, water potential goes down, and water potential in the surrounding epidermal cells is high. So water travels from high potential to the lower potential. So water is also absorbed into the guard cells from the surrounding cells. Therefore, two guard cells are extending, expanding. But this expansion doesn't happen evenly because these inner cell walls are thicker, outer cell walls are thinner. Therefore, outer cell walls are very freely expanding. But you have the microfibril loops which drag the inner wall to the outwards when the cell wall is expanding. Therefore, the stomata opens beautifully like this in the morning. At night, what happens? These potassium ions are again given back to the surrounding epidermal cells. Potassium is given back. Therefore, what happens? Solute concentration goes down, water potential goes up. Because of that, water is also leaves the guard cells to the surrounding cells, surrounding epidermal cells. Water is also released. Then what happens? The turgidity goes down. The expansion is reduced. Now it becomes flaccid. Because of flaccidity, what happens? The outer wall is now reduced, right? Extension is reduced. It becomes flaccid. Because of that, what happens? The inner wall is again coming closer to each other. So the inner wall looks like this. That means it closes the opening. So at night, stomata is closed like this way. This is potassium influx theory. So the energy in the morning for the absorption of the potassium, you need energy because usually the potassium concentration in the guard cells and the epidermal cells are similar. So you have to take more potassium against the concentration gradient. So you have to spend ATP. Now, where do you get the ATP for the absorption of the potassium? That is from the photosynthesis. From the photosynthesis, can you remember the linear electron flow? And there was another one, cyclic electron flow. The electrons which travels from the photosynthesis, the photosystem 2 to the photosystem 1, and some of the electrons goes back and start again from the photosystem, the photosystem 2, or the first leg of the electron transport chain. So from this cyclic electron flow, you produce more and more ATP. That ATP is used to absorb the potassium ions here in the morning. So at night, no photosynthesis, no ATP. So potassium is given back passively. With that thing, solute concentration goes down, water potential goes up. Therefore, water also given back to the surrounding epidermal cells. And the two guard cells become flaccid, and therefore extension is no longer there. And the two inner walls come closer together and close the stomata. Understood very clearly. Potassium influx hypothesis. Okay, right? Before we go to the note, do you have any questions? Did you understand this clearly? Yes, two questions are there. Sir, could you give a brief summary of this part? Which part, Subhisharan? I have missed this question. Sir, Janani is asking, sir, is that H2O comes out means transpiration? No, 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 no. Don't, Janani, that is not transpiration, right? Now, transpiration means water from inside mesophyll cells comes out to the atmosphere. Right? This happens inside this epidermal cell layer. Right? Epidermal cell layer. In the cross section, these are the guard cells. Let's see here. So you have the upper epidermis, lower epidermis. In the lower epidermis, you have the guard cells. Right? Mesophyll cells are there. Right? And right? And transpiration means water is given out from the mesophills to the atmosphere. Make the atmosphere. Now, this potassium and water business happens between these guard cells and these epidermal cells. So, potassium is given to these guard cells. Water is absorbed from the epidermal cells. And again, Water is given back means water is given back, removed from the guard cells to the epidermal cells. To the epidermal cells. ඒක මම ඇන්ද එක පැහැදිලි නැහැ නේ. Tell this one now. So this is the two guard cells, right? Now surrounding this one, you have the layer of epidermal cells, right? All these are epidermal cells, right? So what is given back to these epidermal cells? Okay, understood. Good that you ask any questions. Subishan, from the structure of stomata to potassium influx. Okay, right. Subishan, you don't get it? Structure of the stomata to potassium, right? So I'll quickly explain again. Very rush summary. Import is what I just mean. These, I have to adapt to all these things from the very beginning in Malaysia. Right, I purchased a whiteboard here. It is not fixed parallel. Okay, right. Now, another thing today, your seniors will sit for the exam in August, right? August, and so you will after the exam will be there around somewhere around the 10th of August. So after the exam, that batch is released. So therefore, I also have some enough time, and you also having some free time. So therefore, in the month of August, Pute, we will cover up the whole unit in addition to the regular classes. I mean, classes maybe we'll continue these classes as regularly. In addition, in the month of August, Pute, we'll cover the genetics unit. So then it is easy for us to finish the syllabus earlier. Okay, right? Get ready for that thing also. Cover up finish the genetics unit in the month of August, right? And likewise in December also, we will finish the 10th unit in addition to the regular classes. Okay. Right. So, quickly go there. Now, you have the guard cells like this, right? In the morning, what happens now? You have the surrounding epidermal cells here. These are the other epidermal cells, right? So what happens in the morning? The potassium is absorbed into the guard cells. Potassium, right? Potassium is absorbed into the guard cells actively, right? Potassium taken in. So what happens when the potassium is absorbed into the guard cells? The solute concentration in the guard cells, what happens? Solute concentration goes up. That means water concentration is lower compared to the adjacent epidermal cells. So potassium is absorbed actively, spending ATP, and following that, water also comes inside the guard cells by osmosis. So water also comes inside. So what happens? Because of absorption of more and more water into the cell, what happens? The cell swells. The cell start expanding, right? So the expansion doesn't happen equally. If the expansion happened equally, the stomata will also closed, should be closed. So this one also expanded, no? But that is not the thing that happens because the inner wall is thicker and non-elastic, therefore that doesn't expand. Only the outer wall is expanding, right? It is expanding like this, right? This is expanding like this, and it pulls the inner wall to outside because of the expansion. Therefore, the inner wall is pulled outside like this, and the stomata is open now. This is the inner wall. So this is the stomatal opening is now opened. So that is how the stomata opens in the morning. Understood? Okay, right. So at night, what happens? These potassium again given back to the surrounding epidermal cells. Right? So potassium you give back the potassium. So what happens? Now the potassium is given back to the surrounding epidermal cells. Therefore, solute concentration goes down. When the solute concentration goes down, water concentration becomes invariably higher inside. Therefore, water also leaks from the guard cells to the surrounding cells. Right? Water, like this, it is also given back to the epidermal cells. So what happens? The guard cells shrink like this. So when the guard cells shrink, what happens? The pulling effect of the inner wall is also released. Therefore, inner wall come back, can come closer to each other like this. Because no one is there to pull. The expanded guard cells again becoming shrink. Therefore, stomatal opening is closed at night. Understood? Okay, I hope everyone understood that thing. Subi, are you okay? Ah, good, good. Clearly understood. Happy. Right. Okay, shall we complete the note, Pute? Right. All others are keeping silent. If you have any questions, Pute, just ask anything. Okay. Right. So gas exchange. Now we'll complete this one. Gas exchange. Gaseous exchange. Okay. Gaseous exchange is the exchange of gases between the cells of the organisms. Between the cells of the organisms and the environment. Cells of the organisms and the environment. In plants, in plants, gaseous exchange is possible via stomata, via stomata, and lenticles. And lenticles. Can you remember the lenticles we found in when you learn about the structure of the stems, right? In the cortex, you have some openings, right? Those are the lenticles, huh? So lenticles are placed in the stem, right? So plant gaseous exchange is possible via the stomata and the lenticles. In addition to these, a small amount of gases can be exchanged via cuticle also. Though the cuticle is waxy one, very small amount can be exchanged. Huh? That is a very minute amount. There is no special system within the plants for the transport of oxygen and carbon dioxide, right? So now usually there is a special vascular system to transport the water and minerals, that is xylem. There is a special system to transport the products of the photosynthesis, the sucrose and the glucose, that is the phloem. But there are no tubes or vessels to transport oxygen and carbon dioxide in the plants. But we have our system. Blood is the way that the oxygen and the carbon dioxide is distributed throughout our body. Like blood in animals, the plants do not have a special kind of vessels to transport the oxygen and carbon dioxide. So, how the oxygen and carbon dioxide is transported within the living cells inside the plant? That is completely by the diffusion. Diffusion. That means from the higher concentration of the oxygen is diffused to the lesser concentration cells. That is how it goes. Throughout the plant, that is how it goes. Right? So therefore, these gases move entirely by diffusion. Entirely by diffusion. Entirely by diffusion. Right. So the mechanism of opening and closing the stomata. Now we will write what we learned, right? Stomatal opening and closing depends on changes in turgor of the guard cells. Turgor means pute, the pressure developed inside the cell because of the water. You have met this word previously also, turgidity කියලා එහෙම අපි කිව්වේ, right? So what we meant as turgidity means that turgidity means the pressure built inside because of the water, right? So it if water flows into the cells, if water flows into the cells, if water flows into the cells by osmosis, their turgor, what happens when the water flows into the cells by osmosis? What happens to the turgor? Increased or decreased? Turgor increases. Turgor increases. Just imagine you are blowing a balloon. Balloon එක පුම්බද්දි මොකද්ද වෙන්නේ? Right? You are inserting the air into the balloon and turgidity increases, ne? That the strength is increases, ne? So same thing happens in the cell also. When the water influx, water is absorbed into the cell, the by osmosis, their turgidity also increased, and they expand, but they do not expand uniformly in all directions. Why? Because the outer wall are thinner, inner wall is thicker and non-elastic. Right. The relatively inelastic inner walls make them bend and draw away from each other. This result is the pore, the result is that the pore opens. The result is that the pore opens. Pore opens. If the guard cells lose water, the opposite way, right? So what happens now? What we wrote now, what happens when the water flows into the cell? Now, what happens if the guard cells loses water? If the guard cells loses water, the reverse happens. Other way around happens, right? Their turgor decreases. Reverse happens and their turgor decreases. And their inner walls become straighter, inner walls become straighter, thus closing the pore. Inner walls become straighter, thus closing the pore. The mechanism is explained by the potassium influx hypothesis. This is called as the potassium influx hypothesis. Right? Okay, so this picture shows this is the opening stomata with the influx of the potassium and the water from the surrounding epidermal cells. And this is closing of the stomata at night by releasing the potassium back and giving back the water to the surrounding epidermal cells. Open and close stomata, right? So potassium influx hypothesis we'll write, right? So during the daytime, the guard cells actively accumulate K plus potassium ions, K plus from neighboring epidermal cells, thus lowering their water potential. So when the ions or any solute is absorbed into a cell, Pute, water potential goes down. So you will understand this very clearly when now next we are going to learn about the water potential, right? So pure water is having the higher water, the maximum water potential, pure water, distilled water. When you add salt, magnesium, potassium, anything, what happens? Water potential goes down. So when the potassium is absorbed into the swan, thus lowering their water potential, that leads to the inflow of water. So water is traveling from a higher water potential to a lower potential. So once the potassium is absorbed into the guard cells, water potential goes down, but the surrounding epidermal cells having higher water potential, therefore, inflow of water by osmosis from the surrounding epidermal cells. Thus, lowering their water potential that leads to the inflow of water by osmosis from the surrounding epidermal cells. As a result, Pute, what happened? As a result, turgor pressure in the gut cells increase. So water is absorbed, therefore turgor pressure or the turgidity in gut cells increase and open the stomata. The accumulation of potassium in the guard cells requires the energy. I told you that accumulation or the absorption of potassium from the surrounding guard, the epidermal cells, is an active process. So that needs energy, which is provided by the transfer of electrons during the photosynthesis of the chloroplast in the guard cells. Right? So guard cells, that is why the guard cells are having the chloroplast. So the photosynthesis should happen in the guard cells because they need energy frequently for the absorption of potassium every day. Right? So that is why chloroplasts are placed in the guard cells and the photosynthesis happens. And during the photosynthesis, during the cyclic electron flow, you produce the ATP, and that ATP is, some of that ATP is used for the absorption of the potassium from the surrounding epidermal cells. Okay, right. Then, stomatal closing occurs by loss of potassium ions. Stomatal closing occurs by loss of potassium ions from the guard cells to the neighboring epidermal cells. So it leads to exosmosis. Exosmosis means the reverse of the osmosis, right? That means removing the water. It leads to exosmosis of water from the guard cells to the surrounding epidermal cells. As a result, turgor pressure in the guard cells decrease. And close the stomata. decrease and close the stomata. And there is another thing called abscisic acid. I'll explain it now. Right. Look at this. Now, the basic principle of opening, the mechanism of opening and closing the stomata is mainly by the potassium influx and followed by the influx of water by the osmosis. Now you are crystal clear about that thing. Right, okay. Now there is another small thing. This blue color doesn't go. Right, sorry for the time taken to clean the blackboard. Right. Now, uh, you take a plant, shoot system and root system is there. Right. And now in drought, what happens? In dry season, you don't have enough water in the ground. Right? Very less amount of water is there. So therefore, you can't lose much of the water because you don't have enough water in the ground to replace that lost water. So therefore, you have to minimize the transpiration. One mechanism that is adapted to minimize this transpiration is that from the roots and some of the leaves produce a special chemical. Where does it produced? It is produced in the roots and the leaves. That chemical is called as abscisic acid. Abscisic acid. And you abbreviate it ABA, above. Abscisic acid. Right? So abscisic acid is produced in the leaves and the roots. So basically, the roots know whether you have enough water or not in the ground solution. So when in dry season, when you don't have enough water in the ground, so what they say, they immediately produce abscisic acid and they are sent to the leaves through the vascular system, and leaves itself again as a response, they also produce another set of abscisic acid. What happens? This abscisic acid act on the guard cells, and what they do is they release the potassium from the guard cells to the surrounding uh, epidermal cells. So abscisic acid and goes there and act on the guard cells and they remove the potassium from the guard cells. Therefore, water is also removed from the guard cells. Therefore, the expansion extension is reduced and stomata closed. So therefore, in dry season, very small amount is open when you consider the opening size of the stomata because of the effect of abscisic acid. Just remember that. Understood? Clear? Right. That happens in the dry season. Yeah. Okay. Right. Uh, so abscisic acid, right here in this space, abscisic acid or within bracket ABA also plays a role in the potassium influx mechanism. Abscisic acid, right? ABSCISIC, abscisic acid. Right. Now, the role of abscisic acid in stomatal closure in drought. What happens? Abscisic acid is produced in roots and leaves in response to the water deficiency. And the production of abscisic acid leads to close the stomata by removal of the potassium in guard cells. This prevents the wilting of the plant, right? So drying out of the plant is prevented by this mechanism, right? Finally, right, the last part, this part also we just discussed. Now, what are the factors affecting the stomatal opening? Right. So, four factors will affect the stomatal opening. Four factors, right? The first one you invariably know, right? The stomata opens in the morning and closes at night. So, what is the main intention of the opening and the closing of the stomata is to take the carbon dioxide in and release the oxygen out. So, the light intensity, light, number one factor which opens and closes the stomata is the light. Right? So in the morning or the daytime, the stomata are opened and at night it is closed. The second one is the carbon dioxide concentration. Right. Now, surrounding the mesophyll cells, you have the intercellular spaces and you have the air filled within these intercellular spaces. And the carbon dioxide concentration is low in that inner environment of the leaf. That means you don't have enough carbon dioxide for the photosynthesis. Then again, the stomata opens, right? So when the carbon dioxide concentration is low in the mesophills or inside the leaf, the stomata opens. So carbon dioxide concentration is another factor which decides the opening and the closing of the stomata. Carbon dioxide concentration, right? And anyway, they say, the scientists say they have an inner clock inside the guard cells. That means, it's a common thing that you also should know as biology students. Because we are also having an inner clock. What do you mean by the inner clock? Without looking at the watch, without looking at the daytime, without looking at the light, your body has adapted a way of identifying the morning and the night. Right? So that happens. Now, I am also personally experiencing that thing after traveling from the Sri Lanka to the Malaysia. There is a difference of 3 hours time. Right now, it's around 11:30 here. Right? So Now, still, when I come here, my whole family also, right, doesn't sleep here around. So we usually used to sleep at Sri Lanka around 12 at the midnight. So here now we are awakened until 3 a.m. Still our inner clock is not adjusted to this one. So likewise, every person now assume that somebody is kept in a jail, right, in a cell without looking at the sunlight or the whatever the thing, right? Still, he is getting up in the morning and sleeping at night because his inner clock is working. So likewise, in the guard cells also, they are having an inner clock. So according to that also, opening and closing happen. If you get a plant grown in a pot, you keep that thing inside your home and you close all the doors and windows. Still, the guard cells have the inner clock, so they in the morning time they open the guard cells and at night it closes. So there is a concept called inner clock in the guard cells also, right? And the other one is the environmental stress. Likewise, I told you that in the drought or the dry season, the abscisic acid is released and you try to close the stomata as much as possible. So likewise, the environmental stress, that means in dry season, in high temperatures, right, in high intensity of light, and in the periods where you have a very speedy wind, right? So the wind, temperature, light intensity, dryness of the soil, all these environmental stresses also put a are factors which decide the or which affect the opening and closing of the stomata. That is the last one, environmental stress. Okay, right. These are the four factors that is affecting the opening and the closing of the stomata. Okay. Right. Shall we complete this the part? Hmm. Right. Factors affecting the stomatal action. Number one, light. Number one, light. So stomata opening during the day and mostly closed at night. And light stimulates the accumulation of potassium in the guard cells. Right. So light, stomata opening during the day and mostly closed at night. So that light stimulates the accumulation of potassium in the guard cells. Number two, second factor is the carbon dioxide concentration. Second factor is the carbon dioxide concentration. Carbon dioxide concentration. So decrease in the carbon dioxide concentration in the substomatal cavity, that means surrounding the mesophyll cells, lead to open the stomata. So when the carbon dioxide concentration is reduced in the mesophyll environment, the stomata opens and take more carbon dioxide into the cells. Number three is internal clock in guard cells. Internal clock in guard cells. Internal cell in internal clock in guard cells. So internal clock in the guard cells controlling their daily rhythm of opening and closing the stomata. Right. Number four is environmental stress. Environmental stress. Environmental stress. So, such as drought, high temperature, and wind can cause stomata to close during even in the daytime. Right? Even in the daytime. Okay, right. Now we discussed about the gas exchange, uh, stomata, structure of the stomata, opening and closing of the stomata, the mechanism behind that, and the factors affecting the opening and the closing of the stomata. Right. So we have to discuss the acquisition of the water and minerals and the methods of water transport and the water potential. So that we'll cover up in the next day, right? So any questions, Pute? So this month we'll do 5 classes. I told you the classes, no dates, right? That is shared in the group also. Uh right. So 13th, 20th, 21, 27, 28. Right. Okay. So any questions, Pute? Don't worry that we may be somewhat behind compared to the other classes. We'll catch up this one, right? That we'll catch up this thing. And also, we'll do some complete additional 2 units in the month of August and December, right? In August, we'll complete the genetics. Genetics very important thing. So, most of the part is finished. Questions. No, sir, it's all clear. Right. Wow, great. Okay, so in the absence of any questions, right? So we'll call it a day and have a nice week. Good night, and we'll meet on the next day, right? So anyway, I'll upload the recordings also for the students who missed the today's class. Right. One question came, Subhishana. Sir, your website will be, so in your website, will the previous recordings get expired? If not, it's great help because whenever if there is any doubt can be cleared on time by rewatching the recordings, right? Okay, for your batch, Pute, that is why I am sharing here, right? In the website it get expired, but I am giving you the link which is not expiring, right? So you can see at any time. So this is a special thing that is I am doing for you all because I know you personally. Therefore, I am sharing that thing. That is why I am in the group without hesitation, without looking at your fees payment or anything. I am just sharing. So anyone in your batch can go through the recordings, right? So, so that is not expiring. That is not expiring. And for the students who have not paid the money, and this is their very kind and gentle remind, you can pay the fees to my accountant. Please, the students who paid the amount, the fees, send me the photo or the screenshot of that thing to my personal WhatsApp number. Just a kind reminder. Okay, right. Any questions? No. Right. Okay then. Good night and have a nice week. And we'll meet on the next day. Okay. Bye.